Okay, hello everybody. It is Tuesday night, March 9th. As some of you may have heard, the Wi-Fi at school was out, so I wasn't able to record the lesson, much less post them on YouTube. So I'm back home, and I'm going to make your lesson on what we did today and Tuesday. I have my dog Audie behind me, so he's sleeping, but if he wakes up, hears some panting, or maybe he's trying to push me around, telling me he needs to go out to pee, whatever the case may be. That's what that sound is, but hopefully he'll sleep through this whole lesson. Hopefully you will not sleep through this whole lesson. So today we're going to talk about cladistics and speciation. All right. So cladistics was originally going to be part of the last lesson on taxonomy and phylogeny, but we couldn't fit it into a class period. So this is more or less a continuation of the last lesson. So cladistics is kind of like phylogeny. It's a way to trace evolutionary history, just like phylogeny, but you're using less of the actual data, like evidence for evolution, comparative anatomy, fossil record, biogeography, comparative embryology, molecular homology, and you're going based on traits. So you're looking at the evolutionary history of a group by using shared traits. These shared traits that I have here are known as synapomorphies. If you recall, hopefully you do recall from your Latin, syn means together, and morph means form or shape. So these are common forms that they all have through a common ancestor. Okay, let's continue. So these synapomorphies originate from a common ancestor, and they can be refined and modified as time goes forward. But the basis of those traits or trait are from a common ancestor. All right, the whole goal is to determine, let's rephrase that. This is used to determine which species are most related. Okay? And a lot of this is just done by hypothesis. You're not necessarily going to be right. So I would say this is less accurate than a phylogenetic tree, because phylogenetic trees use the fossil record, they use DNA, they use comparative embryology, all the evidence that we discuss when it comes to evolution. When it comes to cladograms, it's not as accurate. You're making hypotheticals as to how different members of a group are related to each other. You could be right, you could be wrong, okay? But the actual visual that you're going to form is called a cladogram. A cladogram depicts the evolutionary history of a group based on available data. All right. So let's do an example. I'm going to give you guys a list of 7 animals, okay? We have salmon, which is a fish. We're gonna have a frog. We're gonna have a gorilla. We're gonna have a shark. We're gonna have a rabbit, an eagle, and a crocodile. Okay. And I want to know which of these are going to be the most closely related. It's going to be kind of like that game where you all stand up, and I want to go through the process of elimination. So let's say you're all standing up in class, and I say, okay, if you are a sophomore, sit down. If you are a junior, sit down. So let's let their seniors. If you are a senior who rode the bus to school, sit down. All right, so everybody who's standing is a senior that drives. If you're a senior who does not pay your own car insurance, sit down. All right, keep going. If you are a senior who does not pay your own car insurance and did not pay for your own car, sit down. And eventually we're going to get to the individuals that have the most common traits. All right, so we're going to start down here with the very beginning. And I want to know, what trait do you think all 7 of these animals have in common? I'll give you a second to think of that. Don't be obvious like eyes or a heart. You know, that's way too easy. Something unique that these 7 have. Well, it's a vertebrae. They all have a spine. So every one of them inherited the spine from a common ancestor. And now we're going to get to the first outgroup, and I'll define outgroup later. First outgroup is going to be a shark. So this shark does not have something that the other 6 have. What could that be? Now, I don't expect you guys to be experts in animals. Maybe think about it for a moment. What do a salmon, a frog, gorilla, a rabbit, eagle, and crocodile have that a shark doesn't? You ready? A skeleton made of bones. If you didn't know this, sharks have a cartilage skeleton. That's why you never see fossils of a shark. The only part that actually fossilizes are their teeth. But other fish do have bones that make up their skeleton, and those are more likely to preserve. Okay, so we have our first kind of qualifier. Next one, we're going to put the salmon here. Okay. Now remember, this is all hypothetical. You don't know for sure if these are how they're closely related to each other, but you're trying to make a justification. All right, so the next one. What do the remaining 5 have that the salmon doesn't? Give me a second. The remaining 5 are tetrapods. This means they have 4 legs. Like a tripod has 3 legs, a tetrapod has 4. Okay. Next, we're going to have the frog be the outgroup. So what does a gorilla, rabbit, eagle, crocodile have that a frog doesn't? Well, you'll never guess this one. This is you really have to know your stuff. It's an amnion. To briefly explain this, an amnion is a very thin membrane that holds the amniotic fluid, and it also holds the yolk sac. Now, as mammals, we don't have eggshells, but we do start off as a fertilized egg, which is a zygote, and we do have amniotic fluid. When you guys were a fetus developing inside your mothers, you were kind of, I guess, for lack of a better word, floating inside this liquid, and that liquid was contained by a sac, and the sac is called the amniotic sac. That it's made of this membrane called an amnion. If you've ever heard the expression where a woman says, "Oh no, my water broke," that's where the sac ruptures and that amniotic fluid is released. It's basically nature's way of saying, ding, oven's done, time to be born. So now we're going to actually go 2 ways. We have a gorilla and a rabbit. We have an eagle and a crocodile. Well, I'm going to have one trait be here and another trait be over here. Okay. Now, I would not expect you guys to know all these. What I would do is I would give you a list of animals, I would give you a list of traits, and I would ask you to formulate the cladogram. So let's say for these 3, or for the gorilla and the rabbit, those 2, we're talking about fur. So we had the gorilla and the rabbit. You know, you could have said, well, you can't say warm blood because the eagle is a bird and birds have warm blood. Maybe you could have said mammary glands because they're both mammals. They produce milk. Now it could have been a possibility. Remember, this is all hypothetical. And the other one is going to be eggs with shells. Now, if you didn't know this about reptiles and amphibians, their eggs don't have shells. They're like little jelly balls. That's why they have to be in water. And then we have the crocodile, and we have the eagle. Okay, so this is a cladogram. And if we were in class, I would ask you, how many common ancestors do you see in this cladogram? Well, there's one color. There's 1, 2, 3, 4, 5, 6. Remember that as you are going up, time is going forward. So this common ancestor is the oldest common ancestor of all these animals. This is what they all have in common. This common ancestor is the one in which the salmon and the frog, the gorilla, the rabbit, croc, and eagle can all have in common. And it just keeps going. This common ancestor is what the frog, gorilla, rabbit, crocodile, and eagle have in common. This common ancestor is what the gorilla, rabbit, croc, and eagle have in common. This common ancestor is the gorilla and the rabbit's common ancestor, and this one is the eagle and the crocodile's common ancestor. Okay? All right, that is a cladogram. Now, a few other things before we move on to speciation. Let's talk about the outgroup. The outgroup is the species that lacks a synapomorphy. So you could say that in the first round, the shark was the outgroup. It lacked a bony skeleton. Or then you could say the salmon was the next outgroup. It lacked 4 limbs. We have the in-group, which are the species that possess a synapomorphy. So you would say when we had the shark be the outgroup, that meant the salmon, the frog, the gorilla, the rabbit, the croc, and the eagle were in-groups. And then we have clades. Clades are just each species. So there are 7 clades in this cladogram. So we'll say each species in a cladogram. Okay? And that's it. That's cladograms. But now let's get to the other topic I have for you, and that is speciation. Speciation is just the formation of a new species from a common ancestor. So we did this, I think maybe a week or 2 ago. Like actually what makes a species? Whenever I go out on Bayshore Boulevard, I see pelicans and I see seagulls. Well, why is it that what evidence is there that a pelican and a seagull are different species? Well, they really ignore each other. There is such thing as a reproductive isolation, where they really just don't care about each other. I mean, they interact with each other, and I've seen seagulls steal food from pelicans, but as far as mating is concerned, not interested. They're just not interested. But also, when it comes to reproduction, if you have a male and a female in the same species, so here's a male and a female, and let's say they have a little daughter, and then that daughter reproduces, and they have a little son. What we have here is a P generation, F1, and F2. The fact that this individual here in F2, which is the 3rd generation, actually exists, solidifies that these two are the same species. So in order to be considered the same species, you have to A, breed, and B, produce fertile offspring. So how in the world can like the grandson of this couple from the P generation be evidence that the P generation are the same, excuse me, the same species? Well, you have a male and a female, they reproduce with each other, and they produce a daughter. And the daughter was able to reproduce, so that's their offspring. And so that's evidence right there that these guys produced offspring that could make offspring. And so their offspring was fertile. Okay? Well, there's two main types of speciation that you'll need to know. The first one is called allopatric speciation. This is speciation that occurs when two groups of the same species are separated by a geographic barrier. And I have several examples that will hopefully convey this for you. Now, a geographic barrier can be a lot of things. It can be an ocean, or a lake, or a river. It can be a forest, or a meadow. It can be a mountain, or maybe a whole mountain range. It can be a volcano. It can also be some human things like a fence or a road, and even stuff that's really small like a rock. You know, we're talking about ants here. So a few examples. Let's go over one that we've done. Let's draw a little diagram of Central and South America, okay? There's Cuba, Hispaniola, Puerto Rico. Okay, here's the Galapagos Islands out here. All right, well, in this area of Central America, you have the species of iguana called the green iguana. In this area of Central America, you have a species of iguana called a rhinoceros iguana. And then over here in this section of South America, that's Galapagos are technically part of Ecuador, and Ecuador is South America, we have the marine iguana. Well, we do know that members of the green iguana population swam through the Pacific Ocean to the Galapagos, and they swam across the Caribbean Sea to the Caribbean islands. These individuals, for instance, the rhinoceros and the green iguanas, they are reproductively isolated. The rhinoceros iguana evolved in the islands, but its ancestor came from Central America. Well, if you put them side by side in the same enclosure, they're not really interested in each other. They don't want to reproduce with each other. They are different species. And the same can be said about the marine iguana. If you put the marine iguana with either one of these, yes, they're iguanas, but they're just not interested in each other because they are different species. They've been reproductively isolated. Another example could be like this. All right, let's say that we have a lake, and there's this little pillar that's kind of sticking out, and we have these fish species. This is called the red-scaled fish. Well, these fish are able to go back and forth to their respective sides of the lake. They're able to breed with each other, they're able to reproduce with each other, and they're able to just mingle. Well, let's say that the water level, actually drops. Now they're separated, and they're going to be exposed to their own unique mutations, their own unique selective pressures on their corner of the lake. And this is not a matter of if they will become new species, but just a matter of when. And so over time, let's suppose that they actually do become they are reproductively isolated, but they could become different species. And if I were to elevate the water again, and they could actually interact with each other, they're really not going to be interested with each other. They've been reproductively isolated for so long, they formed new species, and now they're not interested in members of the other species. Okay. All right. Well, the other type is called sympatric speciation. Sympatric speciation is speciation that occurs when two groups of the same species live in the same area. So there's no barriers. But they evolve differently until they can no longer interbreed and are considered different species. All right, well, one of the big, so they're in the same area, but they're going to evolve differently. Eventually, they're going to become new species. So let's say that we have some beetles over time. They're going to evolve into new types of beetles. And if I put them together, the red ones and the blue ones together, they're not going to be interested in each other. So these are new species. So how in the world can they evolve differently if they're in the same area? Well, one example is called polyploidism. This is where you have incorrect number of chromosomes. Let me explain to you what that means. And this is very common in plants. So let's say there's a plant species whose diploid number is 4n. Or no, excuse me. Let's just say they're 2n, okay? Don't worry about the number. Well, guys, typically you would have n, and n results in 2n, right? That's what you've been taught. That doesn't happen. It doesn't happen. This is a mistake. Let's say that meiosis goes horribly, horribly wrong, and you have 2n and 2n form 4n. This is known as tetraploid. You know, diploid is 2, tetra is 4. Well, over time, you're only going to have the 2n's interact with the 2n's, and you're going to have the 4n's interact with the 4n's. And that could result in 2 different species. And then you also have something called isolating mechanisms. Isolating mechanisms are what basically prevent viable offspring. You know, for example, a horse and a donkey make a mule. A mule cannot reproduce. So this tells us that the horse and the donkey, the parents, they're not the same species. Same thing goes with the lion and the tiger. They can make a liger. Look it up. They're huge cats. But a liger cannot reproduce. So that means that its parents, the lion and the tiger, are different species. Okay? Well, there's 2 types of isolating mechanisms. There's pre-zygotic, and we'll go over these first. Pre means before, and zygote means, a zygotic means a zygote, a fertilized egg, one cell, something that we all used to be. Well, a zygote leads to an embryo, which leads to a fetus, which leads to a fully formed individual. These are going to be mechanisms in which a zygote does not form. If you can't form a zygote, well, then you can't reproduce. And if you can't reproduce, then you're probably not the members of the same species. Let me say it again. This prevents a zygote, or excuse me, does not form. So if you can't form a zygote, then you can't reproduce. And if you cannot reproduce, then you are likely members of different species. You're not the same species. So there are 5 pre-zygotic isolating mechanisms. Number 1, we have geographic isolation. So you guys have likely learned what a grizzly bear is and a polar bear. Well, they're both bears, right? Maybe they can reproduce with each other. Not happening, because a grizzly is found in North America. A panda is found in Asia. That's thousands and thousands of miles apart from each other. They're never going to meet. They're never going to meet. So that's going to avoid them from making offspring. It's just not going to happen. Another example, you could say is the ostrich in Africa and the rhea in South America. They're both really big birds. They're separated by the Atlantic Ocean, so they're never going to really meet. So that's one way you can prevent a zygote from forming. It's hard to make a zygote, and it's hard to have sex if you can never meet. Number 2 is going to be temporal isolation. And this is all about breeding at different times. Now, it could be different times of the year. It could be different times of the day. An owl and a eagle are not going to breed with each other because an owl is nocturnal, an eagle is diurnal. They're active during the day, owls are active at night. They're never going to reproduce. Also, let's say we have frog species A. It breeds from July 1st to August 31st. And let's say frog B breeds from March 15th to April 15th. Well, they're never going to breed with each other because their calendars are not synced. And so this is temporal isolation. That's all about time. Number 3 is behavioral isolation. This is going to be contrasting courtships. Now, courtship is where you're really trying to form a relationship with someone or something that you're interested in. For instance, with humans, people might go on the first date, they go to dinner, they go out to see a movie, they go for a nice little walk on Bayshore or on the beach, and then they go on a second date, then a third date, then they've been dating for a couple years, then they get engaged, and then they get married, and then they have a family. That's all part of the courtship, where you're really trying to attract someone, a partner, a potential partner. Well, in nature, you have songs, you have colors, you have different courtship rituals. So let's say that you have a cardinal. I'm going to just draw a goofy little cardinal bird. And let's say we have a blue jay. Well, when a male cardinal sings, he is trying to attract a female blue jay. And when a male blue jay sings, he's trying to attract a female blue jay. When a female blue jay is listening and she hears the male cardinal, it's just like, ugh, I'm not interested at all. It's almost as if she doesn't even hear it. And it's vice versa. A female cardinal might, let's say, hear a male blue jay, but she doesn't pay attention to it at all. She's only listening to the song of her counterpart. So members of, when you guys are walking around outside in the summertime and it's raining, and that's what really brings all the frogs out, and you hear all the frogs just chirping and chirping and chirping. They're trying to find a date. If that doesn't get your attention, you go, ooh, there's some frogs out there. Oh, they're looking for a date. That doesn't get your attention because you're not a frog. And so if you were a frog, you would probably jump in the swamp and want to find a date. But behavioral isolation is where you don't have an attraction to their courtship ritual. Number 4 is going to be mechanical isolation. This is where you have a mismatch of genitals, of reproductive organs. The penis and the vagina just don't match up for whatever reason. They're not located in the proper area. It's just not a match. It's difficult to try to have sex and form offspring if the penis and vagina, the reproductive organs, are not going to work with each other. Okay, that can be for a multitude of reasons. Number 5 is going to be gamete isolation. Let's say that you actually pull it off, that you have sex. The sperm and the egg are incompatible. And they will not form a zygote. Okay, so those are the 5 pre-zygotic. Let's do post-zygotic. Isolating mechanisms. This means that a zygote is formed but future generations will not occur. Okay, so first one is called zygote mortality. Let's say the sperm and the egg actually come together and you make a zygote. In this case, the zygote dies. It doesn't even divide once. Number 2 is hybrid sterility. Hybrid sterility means that you actually give birth to the hybrid. When a horse and a donkey reproduce with each other, it's called hybridization. They produce a hybrid, but the hybrid is sterile, which means it cannot reproduce. And that would say that its parents are a different species. And then number 3 is going to be called F2 fitness. This is where the offspring of a hybrid, let's say the hybrid actually reproduces, the offspring of a hybrid are sterile. So for instance, let's say in the P generation, we have a lion, we have a tiger, they form a liger. Liger is going to be F1 gen. And let's say that we get a hold of another liger and they reproduce. Oh my gosh, this has never happened before. And they form a liger. This is the F2 generation. This liger is sterile. It's not going to be able to breed. So, nothing further. All right. Well, there's a little bit more speciation, which I'll get to on Wednesday. But other than that, that's the conclusion of this lesson.